Okay, a very good evening and welcome to our first Royal Society of Biology Southeast Asia branch first event ever. So welcome to this event, and without further ado, our event today is on how do the mechanism behind cell death promote resilient tissue by Dr. Ivan Yu from Singapore. So since this is our first event, so we might want to introduce and invite our chairperson, Dr. Alex Waller, to give a very short speech before we start our event. Dr. Alex, please.
Okay, good evening. And it's good to see so many of us here. Good evening if you're in Southeast Asia, and good afternoon if you're in the UK. So welcome to the Southeast Asia branch of the Royal Society of Biology for our first inaugural event. Less than 9 months ago, I sent a tentative email to MRSB in London asking how many people there were in the ASEAN region who might be interested in forming a branch. And since then, we made a proposal to council that was accepted. So I would like to thank particularly Karan Patel and so many enthusiastic respondents to that initial inquiry, and thank you to the RSB Council for supporting us. In the first few months of this year, we've had a couple of committee meetings, and people have been busy behind the scenes with administration and organisation to ensure we have a wide and stimulating programme of events. So I'd like to say thank you to some people in particular. Without the organisation of Andrew Lucy, our secretary, the coordination and administration of Marissa Koo, our events coordinator, and as well as the social media promotions by Adik and his team, this event wouldn't have got off the ground. So genuine thanks to all of you and other committee members for your support. But without further ado, and somebody else said those words already tonight, but without further ado, let me introduce tonight's speaker, Dr. Ivan Yao from the National University of Singapore, which is ranked consistently as one of the world's top universities. Dr. Ivan graduated with a BSc Honours in the Life Sciences in Molecular Cell Biology from the same university. And after some years of teaching, he returned to the university to complete a PhD. As well as being a member of the Royal Society of Biology, he's also a member of the Singapore Institute of Physics and a research scientist at the Mechanobiology Institute in Singapore. Today, he's here with us virtually to give a presentation on how cell death promotes proliferation of neighbouring cells to maintain tissue integrity. So, for those of you who aren't familiar with Zoom, can I invite you to move your cursor over the reactions at the bottom? And when you click on that, you'll see that if it's the same as my screen, on the left-hand side, there's a little hand clap. So can I ask us to give a warm virtual clap, round of applause to welcome Dr. Ivan Yao.
Okay, thank you very much, Alex, for the kind introduction. So maybe I'll proceed to do the screen sharing. Marissa, I think yes, thank you very much. Okay, well, I'm actually trying to set up my screen. So a very good evening to one and all. So I'm actually quite honored to be the first person to actually do this virtual lecture for the RSB Southeast Asia branch. And as per what Alex and Marissa has mentioned, the topic that I would like to touch on today is how do the mechanisms behind cell death promote a resilient tissue? Now, this topic may be quite foreign to some of the listeners, but rest assured, this is basically very closely linked to what we have learned for cell biology at a high school level and perhaps even at our undergrad level. So do gather the questions that you have during the lecture itself, and perhaps towards the end of it, then I'll collect all the questions and I'll just answer all of them. Okay. So without further ado, let me try to get a spotlight for this. Awesome. Okay. So for the outline of this lecture, I will first go through some background information to get everyone to get into the mood and the context of my research, which I have actually carried out for my 5 years of doctoral studies at the National University of Singapore. And from there, I will talk about some of the gap in knowledge, which is the hypothesis that I actually drew up from my doctoral studies. And from there, my supervisor and I continued to pursue these questions that we have in mind. Next, I'll talk about the methodologies that's used and some of the results that's obtained and what are the impact of this study before I end the talk for today. Programmed cell death, also known as apoptosis, is actually an important role in the development of animals. So this naturally occurring phenomenon was actually observed approximately 180 years ago. So despite the fact that it was being observed 180 years ago, not much is known about it in the field of biophysics. So a lot of biochemistry work has been done. We understand a lot of its biochemistry mechanisms, but for the biophysical parts of it, not much has been known. And this is actually very important because without proper programmed cell death, for the developmental stage itself, when we are actually growing from childhood to adulthood itself, there won't be the removal of these interdigital webs that's found between the digits when we are actually young. And for certain organisms such as the frog, there are certain redundant body parts that needs to be removed. Thus, this programmed cell death is required for the structural deletion of certain parts of the animals that is redundant, such as the tail during its adulthood. So what is programmed cell death, also known as apoptosis? So apoptosis itself can actually be shown in a cascade of events with very distinctive phenomena. So over here, as you can see, I'm actually highlighting this. This is a normal cell. When this cell is poised for apoptosis, it will actually undergo the first step, which is the shrinkage of the cell in size, followed by the condensation of its genetic materials. And after that, you start to see the ruffling of the cell membrane of the cells with the disintegration of the organelles. This blabbing and ruffling will continue to persist until the membrane collapses alongside with the other organelles, and you will form all these small little blebs called apoptotic bodies. And these apoptotic bodies, if they actually persist in your body for too long, it can actually lead to inflammation. Thus, for most of our bodies, including humans, we actually have our immune cells, such as the macrophages, to actually clear them away so that we will not experience any form of inflammation that is unnecessary. Following up from some of the previous literature, this is actually from the lab of Claire Waterman. She has actually studied this process of apoptotic cell extrusion. So when there is apoptosis of a few cells within a tissue, they will actually carry out this process called apoptotic cell extrusion. So in her study, she showed that S1P, which is actually sphingosine 1 phosphate, which is a signaling lipid, it is actually required for the process of removal of these dark cells that are undergoing apoptosis, or from here, I will also call them the dying cells. Now, when the cells are dying, you do not want them to stay within the tissue itself. So therefore, the healthy cells that are surrounding the dying cells will have to cooperate together to form this little purse string structure called the ectomyosin, which is made up of the myosin and the cytoskeleton actin, to actually create this purse string-like structure that will eventually contract and squeeze out the dying cell from a tissue. And this allows the unfit cells to be eliminated from the tissue, and then, as I mentioned just now, let the immune cells within the body do the clearance. So over here, I need to emphasize that this actually emphasizes that other than the mention and the importance of biochemistry within this process, we can also see that the apoptotic cell extrusion is also a mechanical process. So this is actually the work done by my PhD supervisor, Prof. Toyama Yosuke, during his postdoctoral stint at the Duke University. So he was actually a physicist by training, but he actually crossed the boundaries of physics to biology, and he managed to show within the developmental stage of the Drosophila, which is the fruit flies, that during the process of dorsal closure, apoptotic forces were actually required for the sealing up of this dorsal closure gap. And what he did was, very elegantly, he just simply inhibits the process of apoptosis from happening within these particular cells over here. And they realized that when you abolish apoptosis, he measured the forces through traction force, and from there he's able to see that the forces actually decrease, and this actually inhibited, or it actually hampered the process of dorsal closure, suggesting to us that forces are actually important and it's actually coming from the process of apoptosis. So up to now, what are some of the gaps in knowledge? There are two, actually. How apoptosis mechanically influence the neighboring tissue. Because as I mentioned earlier, that Claire Waterman's work has shown that when a cell has undergone apoptosis, the healthy cells around it will actually work together. But how do they know when to start to work together, and how do they synchronize together? And next, what are some of the consequences of the mechanical influence by the process of apoptotic cell extrusion on the neighboring tissue? So with these two questions, I actually went on to pursue this part in my doctoral studies. Next, I would like to share about some of the methodologies that I have actually used within this study and some of the results obtained. So to be able to obtain a high spatial temporal resolution in terms of observing the process of apoptotic cell extrusion, our lab uses a customized UV laser system as shown here. This module is actually attached to a Nikon A1R confocal microscope to allow us to actually pinpoint on the microscope stage which cell that we want them to undergo apoptotic cell extrusion. And by doing so, what we do is we actually use UV laser to melt a portion of the DNA within the chosen cell such that it will suffer a irreversible damage in the DNA, and then it will undergo apoptosis. So over here, published in one of the early papers that we have done with Bernardo Du's lab, we actually showed that this treatment of UV laser treatment is actually so localized that you can see that the damage of the DNA, as shown by this marker, gamma H2AX, is so fine and so concise within this cell. This to show that the UV treatment is not going to cause any lateral damages to the neighboring cells. And next, to be able to measure forces from the tissue or the cells itself, we actually do this technique called traction force microscopy. And this technique is actually rather elegant yet simple. And what we do is, using the conventional glass bottom dishes, we actually fabricate and create a PDMS substrate or gel that is able to be functionalized, and we can actually covalently bind some of the microbeads, which are actually fluorescent microbeads. And then from there, once we have done this, we actually will coat the surface with some ECM proteins to enhance the adhesions of the cells on the top of the PDMS substrate. And eventually we let them grow until they become a confluent tissue. So you can see, this is how it looks like, the glass, the gel that we fabricate, and this is with the tissue. And what we do is that using the UV laser system, we will actually pinpoint a cell of interest to undergo apoptosis. And you can see that from here, we target this cell, and then eventually, after 30 minutes, it will actually undergo apoptosis by showing this blabbing, whereby the dying cell is being extruded apically upwards by the neighboring tissues. So over here, I would like to put your attention to this video. So what happens is that this sea of red is actually the fluorescent microbeads that are on the gel itself. So the asterisk represents the cell that is undergoing apoptotic cell extrusion. We observe this initial phenomenon that is very interesting and not shown by any other labs in the world. And what happens is that from time zero, whereby the process of apoptotic cell extrusion has occurred, we realize that during the process up to about 1.5 hours, there is this wave-like movement of the beads that is radially going outwards in a circular manner from the center of the dying cell. And from here, we are wondering, what could this be due to? We went on to see this, and then we realized that, from the understanding of traction force microscopy, when there is beads movement, we can actually use this technique called particle image velocimetry, PIV, to allow us to actually create vectors. So the vectors itself, we can actually easily understand it as arrows. So the longer it is, the greater the magnitude of the force, and the direction of the arrow tells you which direction the force is actually propelling towards. So we can see that over here, there is this outward propulsion of this outward arrows pointing out, showing that there are forces going outwards. That is actually a representation of what we observed just now from the earlier video, whereby you see this wave-like structure going out. Now, for us to understand if these beats were only moving outwards, we actually did a radial velocity analysis, whereby if you see red means the arrows are pointing outwards away from the dying cell. However, if you see blue, as shown here, it means that some of the beats are moving inwards. And this is actually a characteristic that has been shown by the earlier publications from our collaborator, Bernard Ledoux, which shows that when there is a sea of blue, it is due to the contractility of the ectomyosin ring by the neighboring cells when they are in the midst of trying while the rate outwards movement of the beats is something of interest for us because it has not been observed at all by others from the other labs. So initially we thought, what contributed to the force propagation in the substrate? Now we were thinking perhaps the neighboring cells are able to move inwards to squeeze the dying cell out through cell motility or cell movement. And for that, we were thinking we should do a very simple experiment by inhibiting the movement of the cells, the healthy neighboring cells, by inhibiting this protein called REC1, which is required for the formation and activity of the lamellipodia protrusions. So interestingly, we actually showed here that when we actually inhibited the lamellipodia protrusions, we did not actually affect the actin constriction, which is required for the construction of the ectomyosin ring to squeeze out the dying cell using the purse string mechanism. So next, we went on to look at the beats and we speculated that perhaps if what we speculated is true, the forces were due to the movement of the tissue, then the beats should not be moving at all. Lo and behold, we were very dumbfounded by this finding. After inhibiting the movement of tissues, instead of a diminished or a smaller wave, we actually observe a bigger wave or more enhanced wave.
I think we lost Ivan.
Moving inwards, blue, and the bits that are moving outwards, red. So you can see that for control cases.
Yeah.
So for control cases, we can see that basic
the movement for other side.
Sorry, yes.
They expected like slow movement towards like crushing, right? They actually got a much stronger one from the outside sound and push inwards. You guys hang with me? Those are
Sorry, may I know where did I stop? Is it the previous slide here? Sorry, did I actually stop from here?
Yeah,
no, you stopped at the one before. Like you were talking about how the cells like they had a much stronger reaction. Yeah, here.
Is it this slide? Okay.
Yes.
Yeah. So yes. So what happens is that we observe that maybe I'll off my video first. Sorry, give me a moment. Hopefully this will actually give me a better. Stop my video. Okay. So what happens is that we actually observe that by inhibiting the cell movement, we observe that the ectomyosin ring contraction wasn't affected. So it seems like we have actually removed one mechanism, and we thought that this would also affect what we have observed in terms of the beats movement that we obtained from the traction force microscopy. So from this video, as what I was trying to say just now, and that we were actually quite dumbfounded, because if what we speculated at the beginning was true, and that the wave that we observed was due to the movement of the tissue, then the inhibition of its motion should not give us a bigger or more enhanced wave as shown on the right. So this left us thinking how much enhancement there is. So from a quantitative biology point of view, we try to quantify and see how much has this force propagation actually enhanced. So in normal conditions, the wave actually extends as shown here by the white region, which is the interface between the beads that are moving inwards to the dying cell, and red, which is the beads that are moving outwards of the dying cell. We found this very cool, quiet region, which is the white region here. We define this as the extent of the wave propagation. For control cases or in normal cases, the wave can actually extend up to about 1.5 to 2 rows of normal cells away from a dying cell. However, once we actually inhibit or disrupt the movement of the tissues, we realize that the wave has actually enhanced and extend up to about 3 to 4 rows of neighboring cells from the dying cell. So this is where my supervisor and I were actually very puzzled. Since our initial hypothesis was being overthrown, we actually went back to look at some of the data that we have collected from the earlier works in our lab. So from our lab itself, we actually have worked with other models other than tissues. So Dr. Teng Xiang, his publication in 2017, whereby he works with Drosophila, showed that when you induce cell death over here, demarcated by the red region of the cell, when the cell undergoes apoptosis, there is a movement due to contractility of the ectomyosin. But over here, approximately about 15 minutes or so, he realized that there will be this void region here. You can see dotted lines showing that there is a somewhat detachment of the dying cell from the neighboring tissue. And when this occurred, you can see that these lines that were straight became wavy. And the wavy lines suggest to us that there seems to be a recoil away from the dying cell for the neighboring cells. And this phenomenon was not just observed within the Drosophila model. Another PhD student from our lab, Thomas Mina, in her publication as well, she also showed that using the MDCK model or dog kidney epithelial cell model, during the process of apoptotic cell extrusion, E-cadherin, which is actually a protein that's expressed at the intercellular region or cell-cell adhesion region of neighboring cells, actually decrease or diminish in levels during the progression of apoptotic cell extrusion. And these two results suggest to us that perhaps the false propagation that we observe could be due to the detachment of the dying cell. And thus, we have changed our hypothesis from the initial one to mechanical propagation on the substrate was due to the release of tension in the tissue. So why tension? This is because if you think about it, when a tissue is formed, all the cells are actually adhered to each other. It's like a tug of war that's being held in all directions from a person. The moment I let go of one string, the other party that's holding on to the ropes will actually be dragged by his or her own force that was being exerted by the tension on the rope itself. And this tension itself, we speculate at the beginning when the tissue is formed, the tension is actually kind of like a steady state. It was in a steady state mode whereby the entire tissue is actually balanced out in force. But once the dying cell gets removed from tissue, you're just releasing it, and all the tug walls are being pulled up backwards, and therefore we saw that upward movement. And to be able to actually get some sensing from it, we actually did the PIV analysis on these DIC images from these cells. And maybe if you observe carefully, there is a subtle movement. This is actually a dying cell here. There seems to be a subtle movement of the neighboring tissues once the cell actually undergoes apoptosis. And when we did the PIV, you can see from the arrows that indeed there are this upward movement as well for this level of imaging itself. And to understand, to double confirm that at the basal level or at the region whereby the tissue is adhered to the substrate, if there is really indeed no movement or little contribution by the movement, we actually look at paxillin, which is a focal adhesion protein that is found within the cells when they form adhesion to the substrate. And then we observe the movement of the paxillin speckles with the beads, and we realize that they both correlate in their movement, suggesting that there isn't any form of sliding of the tissue, or there isn't any drastic movement of the neighboring cells away from the dying cell. So to further confirm that this is due to the tissue tension itself, we actually use the UV laser system in our lab, whereby we actually do a fine cutting of this junction, so this black is actually the E-carrying junctions of the cells. Imagine this is one cell, one cell, one cell, one cell. So this is actually 4 cells, and they form this double Y structure. We actually cut a straight junction here, and from there, once it's cut, there will be a recoil moving upwards and downwards, and from there we will actually monitor the distance that is moved by these 2 knots, which is demarcated by the 2 red dots, and we see how fast they move with respect to time. And this gives us a recoil velocity, which is approximate, is actually a very good proximity or measurement of the tension. Take note, we can't use this to exactly measure the absolute tension, but it is a good proxy for tension itself. And from here, you can see that seems like when we inhibit the laminipodia protrusions, or we try to halt the movement of the tissues, there seems to be an increase in the tension itself for the tissues. So for the first summary, what happens is that we have shown through a few experiments to show that tissue pre-tension release was the cause of the propagation of forces. So from here you can see this is the inward and outwards. So if you have a dying cell here, there is a gap here. There will be this relaxation that is pushing the cells backwards. On the other hand, the cells have constructed the ectomasin ring to actually bring themselves inwards to seal up the gap so that they are able to mechanically squeeze the dying cell out. That's why we can see both the red and the blue region for the radial velocity analysis. At the same time, we also look at the changes in the morphology of the cells. So we look at the cells that is directly beside the dying cell, we call them row 1, and cells that are about 4 rows away from a dying cell. From here, you can see that before and after apoptotic cell extrusion, when we follow them through for 30 minutes, there is actually strain or an extension of the length of the dying cell of the neighboring cells adjacent to the dying cells, suggesting that these neighboring cells have undergone a stretch. And we did a measurement and we realized that the stretch is approximately 15%, 15, as compared to the cell that is 4 rows away, whereby the mechanical propagation has not reached to that, and the cells do not actually undergo any form of strain, and that's why you see that it is shown at 0, showing that there isn't any form of stretching. So now we have this big question in mind. We observe this interesting phenomena, so what? What's the biological relevance of this? So we ask ourselves, what consequences of this mechanical influence does it have by the apoptotic cell on its neighboring tissue? Now, we actually did a long period of imaging, up to about 2 days. And interestingly, when we observe events of apoptotic cell extrusion here, we realize that there will be some neighboring cells that will undergo cell division, as shown by these 2 cells highlighted by the 2 red arrows. So you can see, as time progresses, these 2 cells eventually will round up, and after that, they will divide into 2 daughter cells. So next thing we ask ourselves, did extrusion actually promote nearby events of cell proliferation? Our next hypothesis is that the strain that is being exerted on the neighboring cells have affected proliferation in the neighboring tissue. And for that, we actually did some biophysical studies. But before that, we actually used this technology called Fuji. So Fuji basically uses 2 proteins, CDT1 and Gemini, whereby they are actually expressed at different parts of the cell cycle. So CDT1 is expressed using this orange fluorescence. It's expressed during the G1 phase up to the early stages of S phase, while Gemini, which is attached to a green fluorescent protein, is actually expressed around the mid of S phase all the way to the end of mitosis. And during and after cytokinesis, when 2 daughter cells are formed, usually both the fluorescence will not be shown. So over here you can see that this is very nice green-red coloration to show you that the cells are actually rapidly undergoing cycles of the cell cycling process, and they are actually undergoing cell division. By using this technology, we actually created a stable cell line that expressed these 2 proteins with these colors, and we actually did a very long time imaging to observe the events of cell proliferation. So you can see from this video that this cell initially was red, eventually turns yellow. Yellow means that there is this region of S whereby both the proteins are expressed, after which the red is degraded, and then the green will just increase until G2 and M phase. And once cytokinesis is formed, you realize that they have lost the fluorescent proteins. So what we observe is very interesting. We realize that a small number of neighboring cells, I take note, small number, not all the neighboring cells, undergone cell cycling with events near the events of cell extrusion. And we further quantify this to show what are some of the interesting phenomena that we observe. We realize that the cells that are near to the events of apoptosis will actually have a relatively short G1 to S phase transition of about 6.5 hours. Usually it takes about 8 to 9 hours for controls. And we actually counted all the cells and we created the probability of cell division occurring in the neighboring rows with respect to the dying cell. So row 1, as you can see, is the region within the yellow. Row 2 are the pink ones. Row 3 are the blue ones. Row 4 are the green ones. You can see that with apoptosis occurring, there seems to be a significant increase in probability of cell division for the first 2 rows. However, this probability is not increased at all significantly for the 3rd row onwards. So this was interesting for us. Next, we want to calculate the time of division that's required after there is apoptosis events occurring. So if there's no apoptosis events occurring at different parts of the tissue, we follow them through 24 hours, we realize that, well, the events of cell division occurs quite sparsely and randomly at different rows. However, interestingly, if there's an event of apoptosis, the cells in the first 2 rows are unlikely to undergo cell proliferation for about 10 to 12 hours, suggesting that perhaps there's time required for the neighboring cells to prepare themselves for cell proliferation. And this is especially so if you look at the number of events that occurred after 12 hours. You can see that there's actually more dots showing that there's more events as compared to the control. So to be able to link this back to the biochemistry aspect, we actually probe 2 mechanosensitive proteins. One is a pro-proliferative transcription coactivator, YAP, and the other one is actually a marker that is known for the proliferation process transcription, and that is ERK2, that's within the MET kinase pathway. We observe that when there is an apoptotic cell extrusion event, as shown here by the asterisk, the closest neighboring cells will actually experience nuclear translocation of YAP. Nuclear translocation means that the YAP proteins will actually travel from the cytoplasmic region into the nucleus, while the cells that are further away usually will not experience much of this nuclear translocation. And we quantify it to observe for cells that experience YAP translocation. You can see that there is a gradual increment in the levels up to about 3-folds as compared to the normal, as compared to the cells that are actually not near events of apoptosis. So this gives us some confidence that, okay, YAP is known to be involved in proliferation, and there seems to be some response by the neighboring cells. We look at ERK2, and ERK2 itself also shows a similar phenomenon of nuclear translocation, and this only occurs for some of the cells that's near to the sites of apoptotic cell extrusion. We went on to quantify it, and we realized that, well, cells within the first 2 rows indeed have more ratios of cells undergoing this nuclear translocation. And when we went on to probe it further, we want to understand how this behavior is like. And we realized that even though only a small number of cells near the site of extrusion actually have the response for both YAP and ERK2, even the response behavior is actually very different, especially when we can see for ERK2. You can see, as for the few samples, one is a low basal level increment, one is a spike, and one is a gradual increment, suggesting to us that there is some form of inhomogeneity in response. And this gave us the next probe, which I will talk about after the first few summaries. And summarize the next summary I would like to talk about is that we showed that there is a spatial inhomogeneity in cell proliferation. Only a small number of cells actually undergo cell proliferation. And the response of YAP and ERK is also within only a small number of cells, and we are wondering if there's some sort of correlation. Next, we observe a higher frequency of cell division within the first two rows of neighboring cells when there is an event of cell extrusion due to apoptosis. So this is the basic model that we have as of now before we probe another deeper question. So you can see that there is a pre-tension release. It causes a stretch on the cell while they are trying to close the gap due to the removal of the dying cell, and there is a translocation of YAP and ERK within the nucleus. So now, big question here. Where did the inhomogeneity come from? We scratched our heads, we went back to our data, and we looked very, very closely, and we observed that, well, since there is some sort of spatial inhomogeneity, perhaps it came from some factors within the space itself. And we are trying to look for some non-homogeneous factors in space that could help us to define this pattern. And what we did was we went back to our TFM analysis over here. And over here, instead of looking at the outwards and inwards, we look only at the magnitude or the forces where it's actually significant. So if there is regions of the substrate that is experiencing high force propagation, it will be at the yellow region, yellow color region, while if it's going to be low force magnitudes, it will be at the blue region. So if you can take a look at this, we have two cells here that's actually within the yellow region. And these two cells here that are being demarcated here, and the dying cell is the asterisk. After we flow them through for 9 hours,
Ivan, I think you disconnected again.
Can I suggest that whilst we wait for Ivan's reconnection, if anybody has any questions, they might want to type them into the chat. If you look at the bottom of your screen, there's a chat icon. You could start typing some questions now. Ivan,
so sorry. Yes, no problem, no problem. I think today seems to be a bad day because I'm actually on 2 internet connections and both are not working well. So yeah, may I know where did I get cut off? Is it this slide or the previous one?
Yes, this one.
Yes. So over here, I was trying to share that the initial model that we have is that up to now, all the results have shown that stretching of the neighboring cells due to the release of the pretension from the dying cell has actually allowed us to observe 2 proliferative players, YAP and ERK2, to actually translocate into the nucleus that is known to actually help with the transcription of the proliferative genes. So one of the big questions that we have from here onwards is, where did all this inhomogeneity that we have observed till now came from? So we have built up a hypothesis, and that is, since we observe spatial inhomogeneity of cell division, we try to probe non-homogeneous factors within the space or within what we can see from the spaces. So we went back to our data to look at the traction force microscopy data. Now, instead of getting the red and the blue arrows, we try to actually measure forces by looking at the magnitude only. And from here, using this heat map, you can see that regions that experience high force propagation would be in the region of yellow, while regions that have low force propagation will be in the region of blue. Lo and behold, if you see this, there are 2 cells here that are actually demarcated by the white arrows, and this is the dying cell, which is demarcated by the asterisk. When we follow them through for 9 hours, these 2 cells actually undergone cell cycling progression, whereby, if you recall, using the full cheat technology, red to green means that they have actually progressed from G1 to S phase or even to G2 phase. Next, we ask ourselves, what other spatial parameters can we actually use? We observe that the cell cycle progression also correlate with the size of the nucleus of the neighboring cell. So if the nuclei of the neighboring cells have larger areas, they seem to have a higher chance of undergoing cell cycle progression. So you can see this is actually the cell of interest. This is neighboring, and this is actually the dying cell. 12 hours later, this cell, as compared to the other cells, actually undergone an so-called accelerated progression of G1 phase to S or even G2 phase. Now, having these parameters alone doesn't make sense. We are trying to put the parameters together to see if there is a form of combinatorial effect. And we realize that if cells are at the regions whereby there's high force, as shown by the yellow region here, and they have a large area, we actually observe these cells will actually experience the nuclear translocation of the YAP protein. However, this cell, despite the fact that its area is relatively big, because it's in the area whereby there is low force propagation, it didn't experience the nuclear translocation of YAP. So with this inking, we actually went on to probe further. And from here, we actually did a matrix. And from this matrix, we show that for cells that show YAP nuclear translocation, they usually within the quadrant whereby these cells have larger nuclear area, and they are within the areas whereby there is large bit displacement, which is actually a representation of large force propagation. On the other hand, for cells that are likely to divide, they are at the top right-hand corner of the quadrant, whereby these cells usually have larger nuclear area and they have experienced a larger strain, which means that from the imaging itself, you can see the cells being stretched more at an axis. So we're wondering, how does the nuclear area work? So we actually did some stainings whereby we actually use some antibody stains to actually do the counting of the number of nuclear pores on this nucleus. And we realized that, well, lo and behold, as the nuclear size increases, the number of nuclear pore increases, suggesting to us that perhaps for cells that have a larger nucleus, they are perhaps more prepared and more poised for more YAP to translocate into the nucleus. So the hypothesis that we have now is that the mechanical forces that were shown earlier may actually play a causal role in cell compensatory proliferation, in addition to mitogenic signaling. So a bit of background information before we actually show the physical contribution of compensatory cell proliferation. It has always been known in the field that when a cell dies, the membrane is compromised. A lot of chemical components are being leaked out from the dying cell and they spread to the neighboring cells. And they believe that this spreading is through diffusion. And when the neighboring cells take them in, they will then undergo compensatory cell proliferation. However, that hypothesis, that previous knowledge alone is not enough to explain why only a few cells or small number of cells will divide and not all the cells that are close in proximity to the dying cell. So we went on to do more probing, and we want to understand if tissue pretension is indeed very, very important. So what we did was we actually used microfabrication technique to do a stamping of fibronectin ECM proteins, which means that if I stamp a circle that is of a diameter of 100 micrometers, cells can only adhere to this area, and they will form small little tissues, which we call them microtissues. Now, from here, from an engineering point of view, we take it as the smaller the tissue, collectively, the pretension in the tissue is lower. And since we have lowered it, what happens if we kill a cell, as shown in the region by the asterisk? When a cell undergoes apoptotic cell extrusion here, we don't see any form of wave or occurring. You can see from here that the rows, regardless of how many rows are they away from the apoptotic cell extrusion sites, there isn't very strong force propagation. Next, we also went to look, other than force propagation, we want to ask ourselves, are the cells affected even in the biochemical cascades? And we realized that the neighboring cells and cells from it, all of them do not actually experience nuclear translocation of YAP, which again, I'll remind you, is the pro-proliferative transcription co-activator. And we did the quantifications to show that there isn't any translocation. And because of this, there isn't any cells at all within the microtissue that has YAP translocation. And from here, we actually show that the mechanical propagation and also events of cell division is suppressed by the increased adhesion between the substrate and glass. And why do we do that? Because we have known that from an engineering point of view, the gel is actually known to be a viscoelastic material, or you think of it like a very nice jelly that flows like a liquid. So in engineering point of view, if we can strongly adhere this gel to the glass, the gel would actually inherently have the similar property to glass, which means it becomes stiff. And by doing, and to do so, what we did was we did a chemical treatment on the glass surface, which is APTS, that is known to stick the gel, the TFM gel very, very strongly to itself. And then after that, we did the same experiments by sealing the cells and create a tissue. And lo and behold, just by one simple chemical modification, we abolish all the mechanical propagations that we observed earlier, without any form of biochemical response, such as the nuclear translocation of YAP as well. And that being said, we realized that with the coating, even the probability of cell division has decreased or remained significantly similar to that of the control, without different parts of tissue that doesn't have any cell death or apoptosis. And this is actually a very good important information for us, because we have always been talking about the mechanics of the tissue and its importance on cell adhesion, on cell proliferation. And therefore, towards the end of this project, we try to wrap it up, and we see that there is a spatial inhomogeneity in cellular strain. The cellular strain is the one that we observe within the nearest cells that are close to the site of apoptosis. They induce a spatially limited cell division in cells that have two criteria that has been met. Number one, large nucleus. Number two, cells that are actually near to the region of high force propagation. Therefore, we say that these cells actually have a high mechanical stimulation. And by doing so, it actually promotes the nuclear translocation of pro-proliferative proteins, such as YAP, that is mechanosensitive, and this prompts this neighboring cell to be the chosen one to undergo cell division. And the impact of the study is such that we have now we actually gave this new insight to the process of compensatory cell proliferation, because it has been discovered since about 50 years ago, and much of it is not known in terms of the physical aspect. So we have actually added in an additional dimension. And this is important because we know that compensatory cell proliferation gives us the idea that if we can control the transient signaling post-damage or stress, we can actually promote regeneration of, let's say, tissues or even organs. On the other hand, we know that compensatory cell proliferation can be compromised and can be hijacked by the hallmarks of cancer, and this can actually lead to cancer progression. And this gives us the pedagogical relevance to the growth of tumors. And for this work that I just shared, it has all accumulated into two papers, whereby the first one is actually on this topic. It is actually a cell biology paper. And of course, other than that, I mentioned about the physical and the engineering part of the experiments, which we actually work with Tetsuya and Luti, which are actually mathematicians, to actually work on a modeling paper to understand how the force propagation can be channeled from the tissue to this gel and propagate outwards. So with that, I'd like to give the acknowledgement to my supervisor, Prof. Toyama Yusuke, as shown here, with my co-first author for the paper, Assistant Prof. Takumi. And also, I would like to thank my university, National University of Singapore, and the Research Institute that gave me the opportunity with a scholarship to pursue my doctoral studies. And last but not least, I'd like to thank Royal Society of Biology to give me this honorable opportunity to be the first speaker for this event. Thank you very much. I would like to receive any questions.
Thank you, Ivan, to all the participants. So we're going to have a Q&A session now. So I think, Ivan, there are a few questions in the chat box. So maybe you might want to go through it.
Sure. Maybe I will try to take a look at them first. Sorry, give a moment. All right. So, okay, great. So the first question, I'll go from the top. So by Isaac, are there any side effects to the tissue after the removal of the dead cell? So this is a good question by Isaac, because I think I mentioned that earlier studies have shown that there is this mitogenic signaling by the dying cell, which means that there is this diffusion of biochemicals. So what we did was, actually, our collaborators have done this by blocking the spreading of the mitogenic signaling, and we just look at purely the physical influence. And we managed to see that with just the mechanical stimulation, it seems to be enough to actually trigger compensatory cell proliferation within the neighboring cells. So perhaps that could answer partially to your question of side effects if we remove dying cell, the dead cell from the tissue itself. Yeah, or Isaac, you want to follow up on this question?
I forgot to ask, is there any like negative side effects?
Ivan, are you there? Let's give Ivan some time to reconnect back. Sorry for the interruption.
Sorry. Yes, I'm back. Sorry, Isaac, I didn't manage to catch your last question on the negative part.
Are there any negative side effects?
Negative side effects, no. So that means, I mean, and the matter of fact, it's actually good to remove the dead cell, because as I mentioned, in the in vivo system, the dead cells, if they are left there, they will actually trigger the inflammation pathway. So you have this cytokine storm within the in vivo system. But in the in vitro system, because we don't have the immune cells, so if there's a dead cell there, good question. What happens is that if you don't remove the dead cell, that means if this process of cell extrusion is being disrupted, the dying cell usually gets pushed outwards. But let's say if you don't let it push out entirely, it might just retract back into the tissue, and this dying cell, will actually eventually have a chance to become a transformed cell. So that's why for those of us who are actually working with culture, tissue culture, we realize that after a prolonged period of passaging the cells, seeing the cells, you realize that after many passages, you realize that there's a lot of transformed cells. And that could be because of the fact that the cells are trying to get expelled, but at the same time, there isn't a clearance of it. So that could be the side effects. Sorry, Isaac, did I manage to answer your question?
Thank you.
Thank you. Okay, next I have a question from Andrew. So Ivan, you mentioned that the ripple continues up to 3 to 4 rows of cells. Were these studies done in simple epithelial tissue mimics? My answer for that is yes. If so, have you extrapolated as to how this dying cell eviction process and neighboring cell proliferation works in tissues of different types? Good question. So the reason why we're working with epithelial cells, because we know that they have very good, well-established, known mechanisms between the cell-cell adhesion, the cell-cell contacts. So let's say if you're talking about things like the muscle tissues, or you're talking about, let's say, neural tissues, usually their morphology is in such a way that they do not have very well-established cell-cell contact, and therefore I will not be able to actually talk about, let's say, a build-up of pretension within a tissue per se. So that could be a trick. That could be tricky. So I would say that very likely what I observe could actually be more rounded within the epithelial system. But even within the epithelial systems, our lab, before this paper, I actually worked with the MDCK cells, which is the dog kidney cells. I also tried out with the human gastric epithelial cancer cells, and we also tried with other cell lines that are epithelial in nature. And despite the differences from different organs and different organisms, we observe this mechanical wave as well. So hopefully that answers your question. Andrew, is it okay?
Thank you. Yeah, thanks. I mean, I'm just okay. Curious how it would work in a three-dimensional system. That was a primary aspect of it. Because if you're going to be getting a row of cells being moved along on a single cellular matrix, it might have a different propagation process in 3 dimensions. And particularly with other cells that don't have a cellular matrix. For example, and as you say, don't have good cell adhesion.
Yes, actually, if you recall, I can go. Let me call back the slides that I actually showed in the beginning. So my supervisor actually, as I mentioned, during his stint for his postdoctoral stint at Duke University, he actually observed these so-called forces or opposite forces in the in vivo system of the Drosophila or the fruit flies, and that is actually in a three-dimensional system, because I think we all know that for Drosophila they actually have different layers of tissues, and some of the layers of the tissues may not even be living cells, and they have actually very different mechanical properties. So that could also give us a suggestion that perhaps these forces may also be important for the 3D systems, or the, I'll say, in vivo system. Yeah.
All right, great. Thanks, Ivan. Okay.
Thank you, Andrew. Next one, I have a question from Alex. Okay, thank you, Ivan, for such a well-structured. Okay, it was fascinating to learn about the techniques you have employed and how you have been able to build detailed theories from your results. My first question is whether have you studied forces within layers of cells in tissues? Now, to answer that, the tricky part is that, let's say if I have layers of cells within a tissue, for me to be able to track forces, you realize that I actually have to measure the locations of these fluorescent microbeads. So unless we have a way to inject fluorescent microbeads, and I can do a very good Z-stack imaging, such that my mode of imaging allows me to visualize the positions of the fluorescent microbeads within that thick stack of tissues with multiple cell layers, then potentially I'm actually able to measure 3D traction force. So actually this technique exists, 3D traction force, just that usually people use things like a mimic 3D environment like a hydrogel, whereby some cells are actually able to migrate freely, and from there we look at the movement of the beads, and then we can see what are the forces that are being exerted by this motility or the local motion of the cells. So that's the first part. I assume elasticity and shear forces may add additional factors to your model. Yes, and shear forces usually are we usually take them into consideration for models such as the blood vessels, the vascular system. So let's say if I'm looking at the endothelial system, whereby we have the formation of the blood vessels, and when there's blood flow, there's shear stress on these particular cells. And actually, it has been shown in some of the previous works by our institution, MBI. There's a previous prof before she ran over to Canada to take up her associate prof stint. She actually did show that this shear force do indeed stimulate and is indeed able to actually get some response from this shear force, and the cells are actually able to respond to it. So it's quite similar, but they didn't work on the topic of cell proliferation. So they actually probe on other aspects. So perhaps shear force, yes, they do affect, but that will be on the other system. So for me, it's the epithelial system. For that, it could be an endothelial system. My second question is to ask if you know of the applications of your research in treatments such as cancer therapy or inhibiting. Cancer therapy is a big question. I would not dare to touch on that because in Singapore, we actually have another institution, another research center of excellence that is probing questions on that, and that is the Cancer Science Institute, Singapore. But of course, that being said, we do have the links for cancer because people in the mechanobiology field are also doing things like transformation. We actually transform cells by upregulating, let's say, VRAS proteins. And then from there, we observe how would they change when we do different types of manipulation, be it mechanical or be it biochemical. And perhaps this will actually give us some insight of cancer therapy. And of course, now most of us are actually at the theoretical level. So translation-wise, we have quite a big step to go on. As for inhibiting age-related macular degeneration, this is the current new direction by our institution. So currently, we are looking at not just age-related macular degeneration, we're also looking at degeneration or regeneration of the heart. So currently, that's our direction for the entire institution. So maybe you might want to keep track of our research in MBI for this. Yep. Is it okay, Alex?
Yeah, sorry, I couldn't unmute quickly enough. Thank you very much. That's very detailed answers and very interesting news about your research on heart regeneration. Thank you.
Thank you. Okay, and the next question I think is from Marisa, right? As all models are based on animal cells, any chance that this mechanism will also be applicable in plant cells with the presence of cell walls? Answer is yes. Currently, I'm actually doing some very interesting weekend experiments. I actually tried doing this type of experiments on fossils and its effect on plants. And actually, this is actually a nascent field, but there are actually mechanobiology research in the plant itself. And it has been shown that, well, mechanical forces may actually help with certain things like like plant growth. And plant growth, we know that is attributed by, let's say, cell proliferation. So example, if you have a plant that has a root that's going downwards due to the microgravity sensation, we actually have done, there's actually some research has been shown that if you are able to do some sort of a exertion of mechanical forces by pulling the roots with a given frequency, this will actually stimulate the regeneration of the follicle cells within the roots itself, and this could actually help with the plant growth itself. So there are some initial studies going on, and that's for the mechanobiology part of plants. And actually, currently in the National University of Singapore, we do have a 1 or 2 labs that are actually looking at the mechanobiology of plants. So again, if you're interested, I can link you up with that, Marissa.
Oh, interesting. Thank you very much, Ivan. So, okay, right.
Yeah, so for Isaac is Isaac asked again a question. How does a cell lose the ability to start apoptosis? So I think the process of apoptosis requires a few triggers. So I think what we have learned from high school biology is that either you have the chemical triggers, but you have the chemicals. You have the chemicals that can actually induce cell damage to the cells or compromise of the cells, or you have physical damages such as UV, like what we did for experiments to actually damage the DNA sufficiently such that it's not able to undergo sufficient repair. So if you want to kind of like prevent cells from starting apoptosis, the best model to look at is actually the cancer cells, because cancer cells have found a way to not die, but just keep proliferating. So I think going back to what we learned for cancer biology, if you look at all the mechanisms that is being so-called downregulated or being disrupted, usually these are the ones that will give you a suggestion of the importance of apoptosis for the cell's ability to actually so-called bypass the process of apoptosis. Yeah. Is it okay, Isaac?
Thank you.
Okay, so this one is sorry, how do I pronounce your name? Abiose, is it? Did I pronounce your name properly? Sorry.
Yeah, that's correct.
Okay, yeah, thank you for your talk. When the lamellar podia protrusions were disrupted by the RAC1 inhibition, why did that result in a larger displacement of the beads if the upward force of neighboring cells was due to tension release? So this is actually a very good question. So we were really thinking so what we were thinking is this. We know that RAC1 and another protein called RhoA, they are actually antagonistic in mechanism. So when we inhibited RAC1, we are very careful that perhaps RhoA was upregulated in a form of compensatory mechanism. And when RhoA is upregulated, it means that the contractility might be enhanced. That may potentially be a risk, that may potentially explain why there is this increase in strain in neighboring cells, neighboring neighbors. That may be an explanation. But then again, even without, if you think about it, even without RAC1 inhibition, there is still the strain per se. So I can't really give you a direct answer for this, but at least from our understanding, we know that there is this effect of RhoA upregulation or enhancement due to the RAC1 inhibition. So this could be one of the things that we observe, and that could potentially explain why there is a greater enhancement of the force propagation. It could be an increase. So we actually did the experiments. We realized that when you inhibit lamellar podia protrusions, the tissue pretension is actually increased. That could be due to the RhoA enhancement. Yeah, obviously, I hope that answers the question.
Yeah, thank you.
No problem. Okay, Nomi, yeah, is there a way I can access? Oh yeah, I think Marissa has actually answered that. So yes, so I think the recording can be done. Yeah, my apologies to everyone. The internet today failed me quite badly because I tried it yesterday. It was working fine, but I'm not sure why this happens. But I hope everyone managed to capture the message that I'm trying to convey through my presentation. Or of course, if you have more questions, do feel free to direct it to myself, I think through Marissa, if that's possible.
It's certainly possible if anybody has questions to email us at southeastasia The RSB branch, yeah.